Hey guys, we're going to talk about the human genome today, which is an extension of what we've been learning in genetics. So we're going to kind of use the tools and things that we've learned about genes and inheritance and build on that with some other topics. Go ahead and take a quick look at the comic I threw up there for you. It may make more sense here in a few minutes, but hopefully it gives you a good chuckle. So the Human Genome Project, you may have heard about this. It was an ongoing effort to analyze the human DNA sequence. And what a genome is, it's the entirety of an organism's hereditary information. It can be coded in either DNA or, for many types of viruses, RNA. So for us, of course, we've learned about DNA already, so you know for humans we are talking about DNA. And the human genome has over 6 million base pairs, so we're talking about those A's and T's and C's and G's that we learned about a while ago. Many base pairs, so a lot of information. The Human Genome Project was completed in 2003 and it's still being analyzed. The goals were to identify all of the approximately 20,000 to 25,000 genes in the human DNA, determine the sequences of the 3 billion chemical base pairs, store that information, transfer it to related technologies, and then address any ethical, legal, and social issues that could arise from the project. And that's kind of what that comic was leading to, is there are some ethical and social things that people may disagree with. So how do they do that? Well, there's lots of different ways lots of different techniques we can use to study DNA and to figure this information out. One of them is DNA fingerprinting, and that is an analysis of sections of DNA that have little or no known function but vary widely from one individual to another. So this is a picture of a gel electrophoresis, okay, and these bands here are actually marking certain sequences in DNA depending on what was injected into that gel. Now, if you are interested in this, I really do stress you taking genetics next year. You will do some gels just like this one here, and it will make a lot more sense. But for today, we're not going to get a whole lot into that. Another way to kind of look at the DNA of humans or kind of study is a karyotype, which is just a picture of chromosomes as you see here to the right. And we know that this is a human karyotype because there are 46 chromosomes. Notice there are 22 pairs written here, but then there's also a 23rd pair that has an X and a Y. Okay, that could be XY or XX. And some of you may already know, but XY would mean that this human was a male, and then an XX would mean that that was a female. So 46 chromosomes total here, 23 pairs. The two sex chromosomes are the 23rd pair, or the X and the Y in this case, and then the rest are just known as autosomes. So there are some chromosomal disorders. Some are more commonly known than others. This is when you get too many or too few copies of a chromosome. It's caused by non-disjunction during meiosis, and when we get into meiosis, we won't talk a whole lot about non-disjunction, but basically things go wrong during that process of cell division. So some examples are Down syndrome, which is trisomy 21. So if I go back to this slide, this chromosome pair right here, there would be 3 instead of 2, and that would cause Down syndrome. You have Turner syndrome, where there is only an X. There's no Y or other X in that 23rd pair. So this would be a female, but there are many symptoms that go along with it. Some of them you can't really see outwardly, like being sterile, so that one would not be able to have kids. Some can be seen, short stature, webbed neck, drooping eyelids, differently shaped ears, and I'll show you a picture, and if I wouldn't point it out to you, you may not even know that she had anything wrong with her. And then Klinefelter syndrome, you have 2 X's and a Y. So these symptoms would include infertility, language and neurological impairment, gynecomastia, lanky, and youthful build. So I do have a picture of Down syndrome right here. Child with Down syndrome, probably more common to us. And then this girl here has Turner syndrome. A few more that are very severe, especially the Patau syndrome has an extra 13. Many would not survive, but those that did are mentally and motorly challenged. Could be polydactyl, meaning they have extra digits. Heart defects, structural eye defects, cleft palate, spinal defects. Again, I'll show you a picture of that here in a second. Some of you may not like looking at it, but it's not terrible, but it's probably something you may not have seen a lot before. A child with a cleft palate before it's prepared, or before it is repaired, I should say. And then Edwards syndrome has an extra 18. Again, you got mental challenges, low set ears, rocker bottom feet, a congenital heart disease. series, you can read the rest of them there. So this child has a cleft palate. You usually see cleft palate as a repair in people that survive. And Patau syndrome, like I said, is a low survival rate, but some people just get a cleft palate. And it's very, it can be repaired, and you normally see a little scar at their upper lip. Here you see that polydactylism, where you have an extra toe, and this is an issue where you actually have the digits not separating from one another. Alright, so how can we, or what other ways can we use to watch or kind of see the patterns of traits being passed on? And in order to ensure that a trait is actually inherited and not maybe the result of something environmentally, we do have to study how the trait is passed on from one generation to the next in a family. So we use pedigree charts to show the relationship of traits in a particular family, and that can be used to help us with this task. So I do have a picture of a pedigree chart right here. Males are generally represented with squares, females with circles. You'll notice here that they are trying to track color blindness. And the orange indicates colorblind, and the yellow indicates normal. Notice that the female is the only one that has some hybrid versions, means they're carrying the allele, but they're not necessarily colorblind. So why don't we see any hybrid versions in the male? That is because of something called sex-linked inheritance that we're going to talk about here shortly, and what that has to do with is that the X chromosome only carries that allele for color blindness, basically, and males only have one X, so whatever they have on the X is what they're going to have, because their other sex chromosome is a Y. So again, we'll get more into that here in a second. But this is a very simple pedigree chart. I'm going to show you a very complex pedigree chart, and this just happens to be Cleopatra's, and this might be a little outdated. But you can see here that there are some interesting things going on with Cleopatra's pedigree if you follow it. First of all, up here, this female has two male partners that she had children with, so she had two sons. Now notice those two sons got married, and then their kids actually got married and had kids. So we are seeing inbreeding here, which is no big surprise, because in history this actually happened quite a bit. So you can kind of follow that along, kind of see there's some interesting things that go on there. Same thing with the Russian royal family. This year we've heard a lot about the new baby and everything with the royal family. But they have a history or a pattern of hemophilia within their close family, because they like to keep things really close as well. And there are some cases here, I believe, of some cousins getting married, but again, that was in the past. But it does end up keeping some of these diseases prevalent in their family, and you can kind of look throughout here. But hemophilia is a disorder, a bleeding disorder. So if you got a cut, they don't heal up properly, they don't clot as fast as some people do. So again, just another example of a pretty complex pedigree chart, but not all as simple as that example I showed you. Now that other pattern I was telling you about was sex-linked inheritance. Many sex-linked genes are found on the X chromosome only. So for males, a lot of times whatever is being passed on their X chromosome, they are going to get, because they don't have another allele to counteract that gene, per se. So there are more than 100 sex-linked genetic disorders that have now been mapped to the X chromosome. And this, again, reiterating, males just have one, so all X-linked alleles are expressed in males, even if they are recessive. Color blindness is one of those. So if you've probably had these tests before to test for your color blindness. I know there's a few of us in my classes that are colorblind, so you might be able to kind of figure out what type of colorblind you are. There's usually like a red-green, and I don't know, I'm not positive the other one. I know the red-green one's pretty common, so if you had red-green colorblind, I'm not sure if you'd be able to see the 17 in this circle. Alright. We are going to practice this stuff in class because it's kind of complex, but we are going to use the sex chromosomes and actually tie in the alleles of their carriers to figure out how many, specifically daughters and sons, that could possibly be had from certain parents carrying that colorblind allele or other sex-linked traits. So we're going to practice that more in class. I won't get into that a lot right now. And that is it. So go ahead, go back to the video. I know I might have went kind of fast. I apologize for any that I had. I try to keep them to a minimum. But again, go back, get down the notes that you needed. We are going to practice this stuff a lot in class. So I will see you soon. Have a good day.